Okay, we're going to go ahead and get started. Thank you. I am so excited to be here today, really very excited, and thank you for being here. I am Sarah Foye, the founder and president of Team Titin, and I'm an occupational therapist, and I'm speaking to you today as a Titin patient advocate. So changes in the Titin gene can cause a wide spectrum of muscle and heart disorders. The age of onset can range from birth to adulthood, and the severity on impact on health is quite variable. And my family is impacted by both muscle and heart disorders. And that is where my passion lies and why I'm here today speaking with you. So today I'm going to first talk to you about some results of a recent survey of the problems being faced by our community. And then I'm going to tell you a little bit more about our organization and how we are working to address some of these issues for affected families worldwide. I conducted a brief survey of a convenient sample within a closed online discussion group asking the Titin affected community to share what do you think are some of the most common challenges or issues we face related to titinopathies. And now I'm going to share some of the top results with you. And let me guess, if you're working in a neuromuscular clinic, you're also facing some of these similar challenges. The top challenge reported were issues related to getting expert care, and this category was reflected in at least 57% of the survey responses. So I know I'm preaching to the choir, talking to people at this conference because there are so many highly trained clinical experts here at the MDA conference, but it can be difficult to find expert care that is required for ongoing management. Expanding on the care challenges, community members report difficulty getting appropriate surveillance and management for critical aspects of the care, including monitoring their heart, respiratory, bone, or orthopedic health challenges. Many affected families don't have access to good coordinated care, especially depending on where they live and their health insurance coverage. Some people find it hard to get care for other conditions which may actually be dismissed as being related to their titinopathy, which actually might be an independent, unrelated health issue. The next most common challenge they face is a long diagnostic journey and difficulty with getting clarity on genetic test results. And because of this, I'm very excited that we're going to hear from some experts next to discuss good tools to help provide diagnostic clarity for Titin variants and clinical assessment. The community also touched on the psychosocial burdens associated with titinopathy, and this includes feelings of grief, coping with disease progression, and what I describe as advocacy fatigue, which is getting drained from having to continually educate clinicians about rare conditions and needs. So as you can see, our Titin-affected community faces a broad scope of challenges, and to support our community, we founded the nonprofit Team Titin. And our mission is to serve those living with, caring for, or researching Titin-related muscle and heart disorders. And to achieve this mission, we focus on the 4 key pillars of connection, advocacy, care, and research. And having just listened to the keynote speakers and Sharon Hesterly talking about the mission of the Muscular Dystrophy Association, we're very much aligned in our mission. So I'm going to briefly walk through the pillars that are the foundation of our work. The first one is connection, and it's a key pillar of our mission. We aim to connect with and support affected individuals, parents, and caregivers, helping them navigate the complexity of Titin-related disorders. And if you have affected individuals that you come across, please refer them to our group because it's really important for them to feel part of a community and know that they're not alone. We strive to connect also with other organizations and stakeholders that support our mission. So we use various tools to foster connection within our community, including newsletters, online forums such as social media, webinars, YouTube videos, and our website. We also host conferences like SciFam, which provides a platform for individuals to share their experiences, learn from experts, and feel less alone in their journey. Advocacy forms another crucial pillar of our mission. We serve as advocates for our community, amplifying and representing our collective voice, kind of similar like what I'm doing today. This is a very important issue, is while we seek a treatment for tomorrow, we are committed to helping affected individuals access the best care for today. And this commitment to care is the third pillar of our mission. We provide education and information about best care practices for Titin-related muscle and heart disorders and share patient-friendly or family-friendly care guidelines. We understand that knowledge is power, and we believe in empowering our community with the most up-to-date, accurate information about our disorders. And for families who understand what best care practices might be, then they can come to the clinic better armed to interact with their clinicians to understand what their needs are, what testing is needed, and how to access the care that they need. Beyond connection, care, and advocacy, we're also investing directly in the science. And we support, fund, and connect with researchers, clinicians, and other stakeholders. Our goal is to support the search for a treatment and better understanding of titinopathy. We also share research opportunities with our affected community. And so, for example, the kinship DCM study that just started recruiting in February of this year, we would put information about that on our socials and also on our website. And if you have a project that you would like us to share with our group, please reach out. And many of our partners that we share information about are in this room with us today, so thank you. We also work to advance clinical trial readiness, including co-hosting our registry, which is the Congenital Muscle Disease International Registry. We attend global conferences like the one we're at now to learn from and connect with experts like you. And we co-host a biannual case conference and research update virtually. And Marco and I co-host that along with Dr. Bjorn Odd. And these conferences can help clarify complex cases and share scientific advancements with our community of experts. And our next Titin Case Conference is going to be on May 6th at 8 a.m. And if you want to register, there's a QR code on our table that's right outside here in the Advocacy Pavilion. You can click on it and register for that conference. So we'd love to have you. And also, if you have a complex case or unsolved that you want to share with the group, then please reach out because we welcome that. So our research efforts also include grant funding through a peer-reviewed request for proposal process. And our research funding includes development of a skeletal muscle organoid with Dr. Isabelle Richard and her team. We also co-fund research with the Sylvie Family Foundation, supporting Jennifer Rogenbach, who's here with us today, and she's at The Ohio State University, and she works in collaboration with colleagues at the University of Arizona. And her work advances our understanding of monoallelic Titin variants in skeletal muscle disease. And also she has a poster here, which is the poster number there is 331T, so you can check that out and of course find that information in the app as well. And also she's here in the audience, so you could ask her questions. We also co-fund a natural history study with a ZNM charity in Germany with Dr. Anna Sarkozy and her team in London, and you're going to hear more about this project directly from her shortly. So thank you for listening to my talk to provide some perspective as a Titin patient advocate, and you have a sense of some of the greatest challenges faced by our community and some of the actions that we're taking to help support our community through connection, advocacy, care, and research. So now I'm very excited to be hearing from some Titin experts. I'm going to introduce our next speaker. Dr. Marco Savarese is a molecular geneticist with over 15 years of experience exploring the genetic and molecular basis of hereditary musculoskeletal disorders. He leads the myofin research group at Folkasson Research Center in Helsinki, where his team bridges the fundamental myology with cutting-edge genetic and clinical studies of neuromuscular disease. A major focus of his recent work is improving the interpretation of Titin variants, which remain among the most complex and clinically relevant in medical genetics. And I was at the conference at the table, and someone came by our table, and I said, do you face challenges with Titin variants? And they said, I just call Helsinki. So anyway, welcome, thank you.
Thank you. Thank you so much, Sarah. Good morning, everyone. Let me just check that everything works. Yes, perfect. I do not have conflict of interest, and today, for the next 20 minutes, I will go through this presentation, starting with a quick background on Titin, the gene, the protein, the transcripts, to move through the variants that we all identify in our patients, with some closing remarks of what to report and how to report Titin variant. So Titin is encoded by a gene on chromosome 2 with 363 coding exons, and Titin gene encodes for the largest protein in the human body with over 33,000 amino acids. The protein has an N-terminal part in the Z disk, a carboxy-terminal part in the M band, and the protein contains a large number of domains, mainly Ig-like domains and fibronectin 3-like domain. At the end, the protein has a clear, crucial structural function in the muscle, is the elastic, the spring of the muscle, but also through a large number of interactors and interactions, Titin is a crucial signaling hub for muscle and muscle development. When it comes to Titin transcripts, unfortunately, we do not really have a clear understanding on how many different transcripts are generated by this gene. So what we do, and what I will do in this presentation, is just to refer to the so-called exon usage. So I will use the so-called PSI, the percentage splice in, which is a number that measures how much an exon is transcribed. When PSI is high, when it's 1, that means that exon is always transcribed, so it's always included in the transcripts. When it's 0, that means that exon is always skipped. For us, in Helsinki, Titin and titinopathies are a 35-year-old project. Everything started in 1991 when Professor Ruud described the first huge Finnish family where many affected individuals had a mild late-onset myopathies, but few of them had a more severe myopathy with an infantile onset. A few years later, the locus was mapped to chromosome 2, and finally, in 2002, the causative variant was identified in the very last exon of Titin, exon 364. Since then, of course, we have identified a huge number of variants, and we have diagnosed together a large number of patients. And now we will dive through the different variants we may identify. The most common finding is, of course, a single heterozygous, so a monoallelic Titin truncated variant. In the last 15 years, several different studies have confirmed an association between single monoallelic Titin truncated variants and dilated cardiomyopathy. And for truncated variants in Titin, we mean any kind of variants resulting in a premature stop codon. And to be honest, we usually also include splice variants, even if it's not probably 100% correct. However, when we discuss the Titin-related dilated cardiomyopathy, the main issue is the penetrance. We know that the penetrance is not 100%, and we know that if we study families where the probants have an overt cardiomyopathy, the penetrance seems to be quite high, up to 100% at age 70. However, what we did in Helsinki is to study a large European cohort, over 18,000 individuals, mainly affected by rare genetic conditions and their unaffected relatives. This cohort is the so-called SOLVE-RD cohort, so it's part of a large European project. And we reviewed all the Titin findings in this cohort. We identified at the end 100 individuals with exons, with Titin truncated variants in exons having a high cardiac usage. so exons that are actively transcribed in the heart. Of these 100 individuals, only 4 have an overt cardiomyopathy, suggesting a penetrance in our cohort of 0.04. Of course, there is a clear bias here, which is the age, because the average age of the affected individuals is 30 years, and for their unaffected relatives, the age is 44. But to be honest, this penetrance and this number is not really far from the theoretical penetrance if you consider the prevalence of dilated cardiomyopathy and the frequency of Titin truncated variants in highly expressed exons in the heart, which is around 0.506% of the population. But how single monoallelic truncated variants may increase the risk for dilated cardiomyopathy? Well, the mechanism is actually quite complex, because what we know right now is that overall in the heart of patients with dilated cardiomyopathy, there is a reduction of Titin expression. But what we also know is that in the heart of individuals with truncated variants, well, you can clearly see the expression of the truncated proteins. You can see the expression of Titin fragments of the expected sites considering the location of the identified truncated variant. Not only these fragments are expressed, but they are located and they are localized in the sarcomere, where most likely they interfere with the normal Titin function. However, when it comes to skeletal muscle, the situation is more complex. If in the heart we see fragments, which clearly means that Titin truncated variants skip nonsense-mediated decay, in skeletal muscle the situation is more complex. We usually do not detect these fragments. So it seems like for some reason in the skeletal muscle, nonsense-mediated decay acts in a different way than in heart. But what we found is finally this large Belgian family where we clearly see the expression of a truncated protein here. The truncated variant is in the largest Titin exon, exon 3 to 7, and we have in the family a combination of a dilated cardiomyopathy and a mild skeletal muscle disease. Well, to make things even more complex, what we know is that there are not only monogenic conditions where we find single monoallelic Titin truncated variants, but we may have digenic conditions where on the top of the Titin truncated variants, we find a second variant in another gene. The second gene, as reported in this large study from 2024, led by the Newcastle team, by Anna Toff, we have a second gene located on the X chromosome, SRPK3, which somehow is related to the phosphorylation of RBM20, which is the main splicing factor for Titin. Well, this combination results in a phenocopy of Titin congenital myopathies. So in conclusion, when we find Titin truncated variant in heterozygosity, we know that they act as a risk factor for heart muscle disease, but we can't really quantify the risk. But we know that the penetrance is for sure higher in families with a positive family history. We know that monoallelic Titin truncated variants may occasionally cause mild or subclinical muscle disease. I will say mild muscle disease. However, we still think that recessive forms and endogenic titinopathies may be more common than the dominantly inherited skeletal muscle titinopathies. And finally, the full mechanism of Titin truncated variants needs to be fully elucidated. There are clearly differences between the skeletal muscle and the cardiac muscle. The second most common finding is most likely the biallelic truncated variants, so 2 variants in trans. Well, in this case, the diagnosis is quite straightforward. There is a titinopathy, often a congenital titinopathy. There could be few exceptions according to the location, but I would say the most common form of biallelic truncated variants related titinopathy is a congenital form. And when it comes to the phenotype is complex, is very heterogeneous, as you would expect, because there are 363 exons, because the exon usage is different, and so on. I will not go deeper in this, because Anna will focus more on this, but I just want to stress that there are prenatal signs for congenital titinopathies, but above all, I want to stress that in our study, we collected cases from different units. So we involved geneticists, gynecologists, pediatricians, and at the end, what we clearly see is an overlap with other syndromic conditions. We see patients in our cohort with dysmorphic features, with congenital bone fractures, and as reported by our collaborators, by medical geneticists, for some of these cases, differential diagnoses include clear syndromes, like the Escobar syndrome or like the Noonan syndrome even. The most complex aspect is that some of these patients may die prenatally or perinatally, but some others actually have a disease course that is even stable after birth, or actually it improves. And this is for us, this has been for us a big question mark. Why do we see this? Our hypothesis has been that, well, the key is most likely the exon usage. So what we did is to perform short and long read sequencing, and we came up with this tool, which is freely available online, which where for each Titin exon, you can see the exon usage, so how the exon is expressed in prenatal skeletal muscle, postnatal skeletal muscle, prenatal heart, and postnatal heart. And that helped to refine the genotype-phenotype correlation. What we noticed is that the most severe cases resulting in perinatal mortality are those where at least one truncated variant is located in an exon with a very high exon usage before birth. Our hypothesis is that that exon is included in transcripts that are essential for muscle development. So if you have a nonsense there, you do not have those isoforms, and the phenotype will be lethal. Vice versa, we notice an improving disease course in those patients where the Titin truncated variant is located in exons with a, I would say, mid-exon usage before birth, and then at birth, the exon usage of the exon goes down. And for us, this switch here may explain the stable, if not improving, disease course. Just another comment, of course, as you would expect with biallelic titinopathies, most of these patients have a severe cardiac condition, and this is something that should be considered when two Titin truncated variants are identified in trans. So biallelic titinopathies result in a diagnosis of recessive titinopathies. We think the exon usage may help to explain the phenotype, but still we need to better characterize the different isoforms. And then finally, the most complex findings, the non-truncated variants, for example, the missense variants. What do we do with the missense variants? Well, we know that there are two hotspots for Titin variants, missense variants. One is exon 344, where different missense variants have been associated with the HMERF, so a very peculiar, let's say, muscle disease with a severe respiratory involvement. But then the very last exon, the 364, is another exon where missense variants cause either a tibial muscular dystrophy, so when in heterozygosity, missense variants in these exons cause a mild late-onset myopathy, but the same variants in compound heterozygosity with a truncated variants may cause a form of LGMD with an infantile onset. So please recheck variants in these two exons, 344 and 364. What about the other exons? Well, we published last week a paper where we reviewed all the missense variants in Titin with the help of alpha missense. And of course, there are luckily damaging missense variants all over Titin. So what we did is to select a few variants, perform a functional assay together with Professor Gotthel's lab in London, and we clearly see aggregation for a few of these variants. But what we also do is to look for unrelated families sharing the same variants. So we found the same missense variants in unrelated families in trans with different Titin truncated variants, like to boost, of course, the possible pathogenic meaning of these variants. What makes everything more complex? The phenotype, because we can't perform any genotype-phenotype correlation right now with missense variants. So we know that combination of truncated variants and missense variants may either result in a congenital form, but other combination result in much less severe late-onset forms, and we don't know why at this stage. So please remember that there are missense variants, causative missense variants in Titin. There are two hotspots, 344, 364, but please don't forget any other rare missense variants all over the gene, because missense variant Titin may impact Titin structure and function. Let's move now to the final part, what to report in Titin. Well, the first key point is, please, always use this isoform, because this is the so-called meta transcript, and is the only Titin transcript right now that includes all the 363 coding exon. If your lab, for example, use a different transcript, that may result in missing some key variant, because some exons are not included in other isoforms other than that. Well, when you interpret Titin variants, you should consider the phenotype. Let me give a clear example. If you find a monolithic Titin-located variant in a high cardiac PSI in a patient with a cardiac phenotype, well, most likely then that's the cause of the disease, so the diagnosis is pretty clear. But if the patient does not have a cardiac phenotype, then you need the help of a cardiologist, because that's what the guidelines for incidental findings suggest right now. And the genetic risk for cardiomyopathy needs to be calculated with the help of a medical, clinical geneticist, because family history may impact the risk. When it comes to monallelic Titin truncated variant in a myopathic patients, the situation becomes again complex. Why? Because first of all, myopathic patients may mean everything. You don't have to discard Titin, whatever is the phenotype of your patient, because Titin may be related to prenatal myopathies, congenital myopathies, or late onset myopathies. If you find a single monallelic variant, please follow the ACMG criteria, guidelines, perform segregation, because segregation is, of course, an essential criteria. And my suggestion is, before, let's say, confirming the possible pathogenicity of the single monallelic Titin-located variants, look for elusive variants. So perform RNA-seq, perform copy number variance analysis, check other genes, above all SRPK3. And again, if you need an help, contact above all for missense variants, for example, but to be honest, also for copy number variants, because it's a complex topic, which I decide not to focus right now because we don't have time, then contact an expert center. Again, use, please, the meta transcript. This is a crucial point. And again, look at missense variants. Look at 344, exon 344, look at exon 364, and overall, look for missense variants all over the gene, and check other genes. Check SRPK3, for example. But I would say check also RBN20, for example. Go behind the monogenic conditions, because we are at this stage right now. And then a final topic, preconception counseling. We strongly think that if you have a patient with pathogenic variants, which right now mainly means truncated variants, then they should be discussed with patients, and most likely you should involve a medical geneticist. And then that's our opinion. If you have a patient with a monolithic Titin truncated variants, you should involve a medical geneticist, and eventually a carrier screening may be offered to the partner, because the overall frequency of Titin truncated variants is close to 1%. So this is an opportunity right now that could be discussed with a medical geneticist. And with this, I thank you for the attention. I need to really thank Professor Hood on the right, because I said 35 years ago, he started research on Titin, and he started his long research activity in Helsinki. Maria Francesca Di Feo, who is an MD and now completing her PhD, who has been the main leading person behind the most recent studies in our lab. I need to thank the group I have the honor to lead in Helsinki, but above all, I really need to thank all our collaborators all over the world. I need to thank Sarah, because she has done, I think, a huge job in creating a Titin community all over the world. And again, thank you for the attention. I think we will have time for questions later on. So with this, thank you. With this, I can introduce Dr. Anna Sarkozy, MD. Okay, no, it's not me. MD, PhD, and Anna is a medical doctor working in the neuromuscular field for the last 20 years with experience in clinical diagnosis, management, and treatment of, in particular, pediatric neuromuscular disease patients. And she's currently working at the Dubowitz Center in London, a national specialized diagnostic center for rare congenital myopathies and muscle dystrophy. She has been leading the clinical research on these conditions, and above all, in the last few years, let's say, she has been leading a research analyzed to improve the trial readiness for Titin-related congenital myopathies. And this will be the main topic of her talk today. Thank you.
Thank you. Thank you very much for the introduction. I have no relevant conflicts of interest for my talk. Let me start just quickly summarizing what you already heard and how complex Titin gene and Titin gene-related myopathies and cardiac conditions are, and that can involve a wide range of, skeletal muscle conditions, as well as isolated cardiac defects, but also an overlapping conditions with skeletal and cardiac involvement as well. And in genetic inheritance patterns can be also quite complex, with some conditions that can be recessive, some can be dominant, and now we hear also that can be digenic as well. And given the high prevalence of Titin variants in the population, it's also possible that we might see families with so-called pseudo-dominant inheritance as well, and this is something that we have to keep in mind. So the heterogeneity is also evident throughout lifespan, with presentations in prenatal life, with very severe presentations in the fetus, but presentation at birth with the very severely affected newborns, or later in childhood with sometimes milder presentation or more severely progressive forms as well. And then later onset conditions, which are often more selective, affecting maybe a more localized compartment, and of course the isolated cardiac involvement as well. So when we talk about trial readiness, in particular we talk about complex conditions like Titin, it's really important to remember some key points. And I would like to, some of these points have already been covered by my colleagues, in particular the disease characterization and variant interpretation, as well as patient and population engagement, as highlighted by Sarah. And so in my talk, in thinking about trial readiness, I will focus in particular on deep phenotyping, understanding natural history, and in order also to improve optimal care for patients, identify right outcome measures and biomarkers, and finally, how to support achievement of diagnosis for all. So we talk about deep phenotyping. We did have quite a wealth of, quite a number of papers published in the last years, and in particular in the last few years, describing the phenotype of patients with Titin, in particular the recessive form in early life. And I would like to start from this, the more severe end, with what we see in the prenatal and newborn life. And as you heard already from Marco, and there are some features that are very common in these patients, with the prenatal onset of fetal akinesia, with polydramnios, oligodramnios, and contractures that are visible already in prenatal life, and then at birth with severe hypothonia, arthrogryposis multiplex congenita, and often also cardiac involvement, fractures, and severe respiratory feeding difficulties, which sometimes are quite severe and not compatible with life. And as you can highlight, as also highlighted by Marco, many of these patients have recessive variants, majority of recessive variants, and often mostly metatranscript-only variants is present in at least one of the two alleles. It's also important to highlight that often biopsy has been performed in these patients and highlighted a very typical myopathic features, with some common hallmarks being the presence of central nuclei and the coarse multimini cores. And our pathologist, Rahul Pathke, in our lab also described this so-called Sarkam bodies that are quite often visible in these children, with this subsarcolemmal accumulation of mitochondria that is often visible with this sort of peripheral rim, in particular in oxidative staining. But it's also often visible in particular in these very severe early onset infants, is this severe fatty atrophy replacement in the muscle biopsy, sometimes making a collection of valid tissue from the muscles extremely challenging. And this is something that we have observed now in a handful of children in our hospital. Moving on in the lifespan, then we have the younger children and older children as well. And Marco highlighted this syndromic appearance, which is indeed often present with myopathic face, ptosis, but no ophthalmoplegia, which can be very helpful to distinguish from other congenital myopathies. And this Noonan-like appearance, which often can lead to misdiagnosis, an ogival palate, thoracic deformities with pectus excavatum, and then proximal upper and lower limb weakness with thoracic lumbar lordosis, and of course, proximal weakness. The contractural phenotype is also observed both in younger and older patients with joint contractions in the limbs, but also rigidity of the spine and severely progressive kyphoscoliosis that can often lead to need for spinal surgery. And just so to highlight it, as you can see in these pictures, often these children are also quite low weight, and there is often the need for feeding support for these children. The MRI features done in these early onset forms highlight this prevalent involvement of the semitendinosus in the posterior compartment and the vastus intermedius in the anterior compartment and the selective soleus muscle involvement, peroneus muscle in the lower leg. In our clinic, we had quite a number of patients seen over the years, and we recently completed a retrospective longitudinal analysis of about 28 patients seen over the years. The majority of patients presented symmetric proximal predominant weakness with relevant axial involvement. Majority were ambulant, but about 20% were unable to walk, and these were all patients above the age of 2. And while some features were similar to what reported in literature, we have lower frequency of NIV need and arthrogryposis, possibly reflecting the different genetic background of these patients, perhaps with different type of variants and lower number of patients with metatranscript-only changes. We also looked at longitudinal change over time, and we have in particular looked at motor abilities, respiratory and cardiac function in these children. While there is somewhat stability of the 10-minute walk test in ambulant patient and somewhat stability in the respiratory function in non-ventilated children, if you look at the fractional shortening, you can see that there is a somewhat deterioration over time, in particular in teenage years, and more investigation clearly is ongoing in the prospective studies. And when we talk about younger patients, important again to remind about the digenic form of the SRPK3 Titin myopathy, which really the currently available information really highlight the overlap and the clinical similarity with the classic recessive biallelic titinopathies that is present also on the MRI pattern of these children with this typical semitendinosus involvement, and also the muscle histopathology of these children, which again highlights the presence of central nucleation, of course, and mini-cores, making sometimes a distinction clinically and histopathologically on MRI basis between the classic biallelic titinopathy and the digenic form, sometimes quite challenging. So as Marcus said, please don't forget to think about SRPK3 as well if you have patients with single titin variants. For sake of time, I will not be able to go into major details, and you already heard Marco mentioning about the adult onset, mostly dominant forms of myopathies associated with Titin. Just a couple of quick comments about, in particular, the so-called HMR form that was originally described in a couple of large kindreds from the north of England. And this form has associated with severely progressive respiratory failure. That really is something that has to be, it's important to recognize and treat quite rapidly in order to avoid more severe complications for these patients, but also the absence of relevant cardiac involvement in association with this form. And typically patients present with myofibrillar changes on muscle biopsy, which is somewhat different from what's seen in the recessive forms. And then, of course, the tibial muscle atrophy, which is mostly lower leg prevalent with relevant weakness and atrophy in the lower muscles, but myopathic changes on biopsy, and more prevalent involvement of muscle MRI in the lower leg. And without making too much comments on this, but as Marco said, now we also understand that there is also this axial myopathy form. And again, we can recognize these specific MRI changes on the muscle MRI with fatty fibrous replacement of the paravertebral muscles on MRI. Finally, but not the least, I mean, clearly we have to make some comments on the heart. I'm not a cardiologist, but clearly cardiomyopathy is one of the most common effects of titin truncating variants, but we also understand that prevalence can be different, and as Marco said, there might be, the prevalence could vary in lifetime, but also depending on sex. So males usually have earlier onset, and there is also now this hypothesis of the second hit that is necessary to to increase penetrance and clinical outcome for patients carrying truncating variants, and also the importance of prophylactic intervention. But as you heard, it's very likely that this is, cardiac and skeletal condition, perhaps not completely isolated entities, and there is more evidence now gathered also from MRI and the muscle biopsy works, highlighting that there is a somewhat continuum between this condition. So it's always important to keep in mind both heart and muscle when looking at Titin. Epidemiology. So we know that Titin variants are the most common cause of dilated cardiomyopathy, but what about skeletal muscle? So there is not much clear data on this. We have recently completed a large analysis of over 1,600 UK patients referred to our center, and Titin really is one of the most common causes of early onset congenital myopathies or congenital muscular dystrophies, being the fourth most common among all the patients referred, and the second most common form of congenital myopathy. And this is probably a likely underestimation, because we have, as you can see, a very high number of patients with single Titin variants or variants of unknown significance. So further analysis of these patients might increase the number of patients diagnosed with titinopathy. So we believe that really it's very likely Titin being one of the most common forms of congenital myopathies, in particular the recessive titinopathy. What about in other populations? So we know that there have been some mentions of similar works done in Australia, but interestingly, as part of an international collaboration, as part of the ICGNMD consortium, we have completed analysis of over 100 patients with congenital muscular dystrophies and myopathies, and we have found no Titin variants. So why is that? Could that be because the prevalence is different, or perhaps there is maybe less, maybe titinopathies are less recognized in this population? So this is still unknown. Okay, so in the world of trial readiness, really optimization is care is important, in particular in absence of cure, and understanding natural history is important to understand how we can identify the best intervention to delay onset of complication, and possibly also hoping that one day there will be a cure to completely stop the progression over time of the condition. And for this, it's important also to follow international standards of care. And Sarah already highlighted the availability of guidance for congenital myopathies, and we hope that in future we can also develop standards of care specific for titinopathy. And clearly, natural history knowledge is key to fully understand what is the best standards of care for these patients. And this is what we're trying to do. And we have now started this first natural history for recessive titinopathy in the UK, with the aim to describe not only the natural history, but also to identify the best outcome measures and tools to detect change over time, to try to also to help trial readiness, and also to validate imaging to support diagnosis, drug disease course, and predict outcome. The study started in 2024 in 2 sites in London. We recruited 21 pediatric patients, and we are doing 6-monthly assessment of medical physiotherapy investigations, a respiratory assessment, and 12-monthly imaging. I will present a few of our data analyzed so far. We have a battery of functional outcome measures and motor time tests performed, which is similar to what is ongoing for a parallel natural history study. We're also including focus on fatigue, also with self-reported questionnaires, and the results to date show that these tests are feasible, and fatigue does not seem to affect the functional test. And again, this highlights the importance also to consider patient-reported outcomes when doing this type of studies. Further analysis is in progress for the 12 and 18 months data, which seems so far to show some stability over time of this course. We're also looking at respiratory assessment that has been completed so far in 16 patients aged above 5 years. And again, you can see quite a relevant progression of FVC abilities at 6 and 12 months, with the gray bars showing patients with an FVC below 60% that has been increasing over time. And also the frequency of chest infection hospitalization is similarly increasing. We have now calculated a change over time of the FVC predictor of 3.4%, which is quite important. We hope that this data can help us also to identify if these could be good, valid outcomes for future clinical trials. And we're also looking similarly at progression over time in muscle ultrasound that has been completed in 20 patients. One little girl wasn't keen to have some gel on her legs, so we didn't do that, but all the other patients were happy to perform. And the ultrasound is very feasible at bedside and highlighted a clear pattern, and with in particular some muscle compartments being more affected. And overall, there is some stability on muscle ultrasound over 12 months, and we are now going to compare this with the functional assessments and also with MRI. Just wanted to highlight that similar studies have already also been completed in other centers, and the group from the NIH have presented similar work at the WMS a few years back, and they highlighted typical changes, in particular in the biceps and the triceps muscles. So again, many other centers are focusing on this analysis as well. We're doing MRI as well, and we have done it in 8 patients and repeated at 12 and a 24-month interval as well, again looking for change over time and see if this can correlate with a possible outcome. In the meantime, we are also completed the large collaborative analysis on 48 patients with recessive titinopathy in order to identify patterns and see if there is any difference with patients with different mutations, truncating or missense, and this work is being finalized now, but again, other centers have focused attention on these points, and the pattern of muscle involvement that we notice is very similar to what also described in other publications, different, however, from what's seen in HMERF and Titin and distal myopathies, clearly where they're more, in particular in the distal myopathy, with more localized lower leg involvement is noted. So final couple of slides is for how to achieve molecular diagnosis for all. And you heard already Marco, who gave you very good hints about how to approach this. But I just wanted to highlight that really is challenging to diagnose titinopathy because the presentation, as you heard, are so complex and not unique at all. And there is so much overlap with other conditions. And sometimes it is challenging. So what to do if you have a suspicion of titinopathy? Clearly, we all go genetic first, but just please remember in particular in hypotonic infants to exclude common condition, and don't forget to do the classic tests like exclude, do a CGH array or exclude, SMA, in particular for conditions that could be treatable. So exclude those. And then if you do genetic first, always consider an MDT approach to the results you have, and try to put all of these into an MDT context with involvement of specialist center and research colleagues as well. So clearly, if you find pathogenic variants and this segregates well in the family, your diagnosis is confirmed. But when you don't have, as Marco highlighted, do further investigation, consider doing imaging and muscle biopsy to support your diagnosis and suspicion for titinopathy, and consider other investigation when you are missing the second variant or if your variants are of unknown significance. And again, this often is done only in research environments, so collaboration with a specialist center is key. And we have indeed done our RNA analysis in 22 of our patients where a second variant was missing, and this was very helpful. More than half of our patients has been diagnosed, and we have now publishing these results, so hopefully this can be out soon, but really RNA has been so helpful for our patients, but also to maybe identify SRPK3 variant that we're missing. And we believe that the higher diagnostic yield in our cohort is because we selected the patients based on a very high MDT approach, and because we started from muscle, and because we did total RNA sequencing in collaboration with the group of Marco. So really, this was a great added value. And really just to highlight again how all of this cannot be done if you don't have ongoing research collaboration to really help to evaluate the role of these changes. So my last slide, just to summarize, I think it's quite obvious that titinopathies are very complex, but we are learning more, and hopefully we are a little bit closer to trial readiness. In the treating effort, we are now planning new steps. So we want to finish data analysis and correlate results, but also looking at new aspects like exon usage in the muscle. Could this explain the variability that we see in progression over time? Look at heart, bone, and also think about expanding further to new cohorts and new countries. And we're very happy to consider collaborating with other centers. We already started discussions with Leslie Hayes from Boston Children, but we're happy to share our protocol if anybody would be interested to collaborate. And really, there is the need to collaborative efforts to identify best outcome measures and understand better which are the best treatments for this condition. And finally, I would like just to acknowledge all the team in London that is helping to move forward in this trial readiness path, and all the collaborators internally and externally from all around the world, and our founder, Sarah in particular, and all the families who are part in this journey. Thank you. And I think we are open for discussions and questions.
If you will forgive me as the pulmonologist in the room, for folks who can't stand, FEC percent predicted is highly inaccurate. Estimating height is really pretty much a guess in folks who are non-ambulatory. I think our best estimates using things like ulnar length or knee heel were probably at least 5 centimeters off, if on average, if not more. I'd like to plead with you for a couple of things. One, to look at absolute values of FEC and look at how that changes over time. And two, being that you're doing this beautiful natural history study, maybe we can get some better ways to estimate standing height in the folks who are still able to stand. My gut feeling on this is that it takes several measurements, not just one body measurement, that if we include 3 or 4 measurements, is a tibial length, ulnar length, arm span, demispan, you know, the one arm, measures within the hand, things like that, and we combine them all. Can we get some better measurements over time? And should we be looking at measures that are specific to our neuromuscular or population, as opposed to things that were validated on a bunch of young dancers? But thank you much.
Thank you. You are right, and just to highlight that in our, I didn't because of limited time reason, we are also looking at the absolute values for the FEC. We're also looking at MIP and MEP and SNP in these patients, so it will not just be the FEC predicted. Majority of our patients in the study are actually ambulant, so we have their height is measured by height, but clearly there are patients with contractures as well, so you have to consider also the effect of contractures or on your total height. So these are all relevant points, and we are going to look into all this. Thank you.
This one's from an online member, and I believe this one's for Anna. Is this the same group that has a lot of respiratory weakness? The same group that has a lot of respiratory weakness. Is it the same?
Yes.
Yes.
HMRF. So the HMRF is relevant, is characterized by this very severely progressive, rapid onset of respiratory involvement in adult life. That's the question. But generally speaking, titinopathies characterize in particular the HMRF, and the early onset titinopathies with relevant respiratory involvement, both in pediatric age and progressively later on, there's progressive change in respiratory ability. So I'm not sure if I understood the question.
Yes, thank you. I want to say that I think it's fantastic to have Titin section for Titinopathies here. And I do have 2 questions. One is related to mutations that we see in the PVK domain. What's the clinical manifestation when you see there, especially because it's a domain that changes between heart and skeletal muscles? PVK. And my second question is that, considering that Titin, 363 exons, do you see a chance for exon skipping therapies within that?
For PVK, you mean missense variants or what kind of variants? I mean, for truncated variants, I will say right now, I mean, you should consider all the kind of truncated variants wherever they are located. I will not really, or we do not have such a clear genotype-phenotype correlation that we could really make an assumption on that. For missense variants, the situation is tricky. We have validated few of them, but we don't have data from large studies on that. So I can't really answer your question if PVK missense variants may have, I don't know, a more damaging effect than other variants. And the second question was on gene therapy. I think exon skipping. This is tricky. I mean, I think first we should get a clear idea of titin isoforms in different skeletal muscle and during development. I think for us, that's right now the most urgent thing. I have the feeling that RNA-seq, long-read RNA-seq will help, but you should consider issues due, however, to the size of Titin, and that can include or add a bias on that. We are now, I think it's out as a preprint, but we clearly show that if you perform poly-A sequencing, a poly-A selection versus ribodepletion, for example, your results in terms of your exon usage on Titin will be totally different. And this is a bias due to the enrichment, the transcript enrichment, the MRI enrichment in poly-A versus ribo. So there are technical issues in that, but I think that's the most urgent stuff right now, to first have a clear idea of the exon usage and isoforms, and then maybe, hopefully, we could consider playing, let's say, with exon skipping as a form of therapy. Yes, please. Yes, this is true. I agree with that, yes. In that sense, the location of the variants may make a big difference. I agree with that, yeah.
So they did follow up. They said you were talking about the axial myopathy and scoliosis group at the time.
Yes.
Axial myopathy, yeah.
Yes, so the axial myopathy is, as was recently described by the group of Marco and patients with this is quite a complex condition. So Titin is not the only cause of axial myopathy, and it's probably all the genetic or non-genetic cause of axial myopathy are not fully understood. But evidence is that Titin variants can cause this selective axial weakness, in particular with onset in adult life. But again, axial weakness is part of the phenotype of early onset titinopathies as well. And patients very often have axial weakness and scoliosis and respiratory involvement that is progressive over time.
Hi, Matt Wheeler, and I'm going to represent the cardiologists of the world. So first of all, fantastic overview from both of you, and really learned a lot. I think very excited to see some of the work in RNA-seq. Certainly, there's lots of intronic variants as well in Titin, and so adjudicating all of those is an additional challenge in addition to all of the missense variants. I was wondering, there's a relative paucity of structural variants discussed. Is that something that is a detection problem, or are they really missing from the compendium of patients?
Yes, structural variants, yes. Yeah, we didn't have time to focus also on that, or to discuss that. We have an increasing number of copy number variants right now, going from single exons, deletions, or duplications to much larger deletions or duplications. My personal feeling is that if there is a deletion and that's an in-frame deletions, then most likely I would expect disease to be dominant, and this is what we have observed in a number of patients so far. What is very tricky is that maybe this is not really true for all the exons. We don't have right now a clear understanding of what can be tolerated in terms of deletions and what is absolutely non-tolerated right now. But what we have also identified right now is larger insertions or larger rearrangements, for example, in a Russian patient. And those are tricky. And that has been identified by long-read sequencing. I think also Anna has a similar experience on that. So I would expect now with the more routine use of long-read sequencing to identify more complex structural variants in Titin as well. How to interpret them? Luckily, we are not there yet. We will try to face that issue when then we have a larger number of structural variants or complex variants then. Thank you for the question.
I have two questions. One of them, you mentioned that in congenital autosomal recessive titinopathy, you need to do muscle biopsy to confirm the diagnosis. Is there a specific pattern in the muscle biopsy in those congenital autosomal recessive titinopathies? That's my number one. Second, for MRI in patients, in children, we have, for the clinical purposes, we have two concerns. One is insurance coverage. The second is sedation before age 6. Do you have any patients before age 6 that did MRI, and do you use general anesthesia for that?
Okay, so first thing I wanted to mention that if you have a clearly pathogenic variant and your diagnosis is solid, you don't have to, it's not that I don't want to indicate that muscle biopsy has to be done, but in some cases it's necessary in order to confirm the suspicion or also to get tissue for further investigation. So nowadays, despite the fact that we have genetic testing being so much more widely available, biopsy is still a very important tool to confirm your diagnosis and to do further investigations. And the biopsy, when you do a biopsy, clearly having a histopathology that is supportive of diagnosis of Titin is very important. Or otherwise, you can also identify markers that could maybe indicate a different diagnosis and maybe highlight, actually, this is not titinopathy, but it's a muscular dystrophy or a neurogenic condition and so on. So there might be other findings. So that is one thing. But we don't do it routinely for everyone, and we have quite a number of patients.
But there's no specific finding for autosomal
So the finding is myopathic changes, like in the recessive form, it would be mostly central nucleation or internal nucleation and coarse and multimini coarse that is very often seen in majority of these patients. So these hallmarks but these are not typical and only for Titin. These are commonly seen in other similar myopathies. Although, again, there might be some features that perhaps could be more, sub features. But anyway, yes, so there are some features that are more common. MRI, so all the patients we are indicated here all were above the age of 5 and all had MRI done without sedation, fully awake status. I'm working in the UK, so we don't have insurance problems, and this is part of their clinical investigations. But I would hope that particularly if you have an undiagnosed patient, that this is part of a diagnostic process for a patient, and there is really an added value to identifying the patterns on MRI. But if you had to sedate or do a general anesthetic, clearly, given also the fact that MRI will not be sufficient to confirm or exclude titinopathy, clearly you would rather do a biopsy or other investigation. Okay. But we don't do it on their
younger patients. Okay, thank you.
Yeah, first, just to amplify what Matt was asking and say what you were saying, I think, is that the paucity of structural variants that we know about really is a bias of ascertainment. And as we do more long-read sequencing, that number is going to rise. But Marco, I have a question for you about identifying non-pathogenic missense variants. I think I recall a few years ago you had talked about looking at phased missense variants in trios. So if you have a parent who carries one truncating mutation, for example, and is healthy and has passed that on to a child who has a biallelic disease, can you look at those missense variants on the other allele?
Thank you so much. This has been, as you know, this has been for me.
How has that been going, and can we all contribute?
Yeah, I want just to recap the idea. So I always thought that main issue for Titin is that we do not have a list of non-pathogenic variants. And if you think about that, that's challenging. Of course, there are non-pathogenic polymorphic variants, but that's easy because they are very common. But what about very rare non-pathogenic variants? How can we select them? And they are needed for functional studies, for validating our in silico tools, and so on. So one suggestion I had a few years ago, and it was a topic of an ENMC meeting, is that, well, if we consider the parents of our kids with the congenital titinopathies, they may provide useful data in terms of missense variants, because considering a kid with 2 truncated variants, each parent has 1 truncated variant, and on the other allele, the parent will have missense variants. We can't really say that those will be benign variants, but at least we can for sure say that those variants in compounded heterozygosity with the truncated variants do not cause a severe congenital titinopathy. So my idea has always been that if we are able to really put together this in, for example, a free open access databases, that will really help the community. I know there are legal issues in that eventually, and this is not always the easiest, but we really need sharing in the field. I mean, for Titin, there is, I think, nothing else than sharing. We have to find a way for that, but...
Just to add on this note, that is also further complicated by the fact that many laboratories might not report a non-pathogenic variant or variants of unknown significance in the UK that sometimes, diagnostic laboratories would reach and put it in the report. So it's complication.
We should revisit doing this internationally, and I don't think there are really any privacy considerations if all we're doing is sharing
I agree.
a VCF file of just variants within the Titin locus.
I agree with you, but as you know, not everyone agrees with us.
I just wanted to mention to you, I know there's a cardiologist in the room, that there is a free genetic testing for people with non-ischemic cardiomyopathy that's now available called Heart in the United States, called Heart Gene, and the information's on our website. And also, this came up in our last scientific family meeting, which was Peter Hackman coined that phrase, sharing is caring, and people really like that. So I say, I think it is a really nice international collaborative group, and so please invite you to join us at our scientists, clinicians invited to join us at our next case conference. You can go to our table in the exhibit hall to register, and if you have issues, to connect to this team. They're extremely collaborative, and so I really am grateful to the Muscular Dystrophy Association for having this meeting. We're really happy, and I'm very grateful to our speakers, and thank you for attending. It really means a lot to us at Team Titin, so thank you.